For decades, science viewed aging as a one-way street. The assumption was that getting older meant an inevitable accumulation of physical wear and tear, much like a complex machine slowly grinding itself into a state of permanent failure. A new framework in regenerative medicine argues that this mechanical model is incomplete. Instead of physical hardware damage, biological aging is increasingly understood as a loss of the critical data that keeps our cells functioning properly. This data is managed by epigenetic markers. These are the chemical tags that sit on your DNA and act as a set of instructions, telling a generic cell whether it should function as a heart cell, a skin cell, or a neuron. Over time, these chemical tags become disorganized. This layer of epigenetic noise makes it harder for the cell to read its own instructions, leading to a loss of metabolic efficiency and specialized function. But there is a catch. Beneath all of that accumulated noise and static, the original, youthful DNA code remains perfectly intact. The cell is still holding on to a hidden, pristine backup of its original information. This means longevity science is no longer focused on simply patching up physical damage. The new goal is to develop molecular tools that can clear away the noise and restore youthful gene expression across the entire body. This concept was first proven in 2006 by Japanese researcher Shinya Yamanaka. He discovered that by introducing 4 specific proteins, now known as the Yamanaka Factors, he could reset an adult cell all the way back to its embryonic state. The discovery earned a Nobel Prize and established the foundation for modern cellular reprogramming. But it also presented a major safety risk. Resetting a cell to an age of 0 erases its identity. If a heart cell or a skin cell is pushed too far back, it stops performing its specific job and begins to divide uncontrollably, forming dangerous, fatal tumors called teratomas. To solve this, researchers like Juan Carlos Izpisúa Belmonte pioneered partial cellular reprogramming. Instead of a total reset, they exposed cells to the Yamanaka factors in a brief, controlled burst. On this timeline, you can see the cell moving back toward a younger state without ever reaching the dangerous embryonic stage. This transient exposure is enough to scrub away the age-associated epigenetic noise. The cell's gene expression is refreshed, but it safely maintains its specialized identity as a heart or skin cell. By identifying this specific middle ground, scientists moved age reversal out of the realm of lab theory and toward a practical medical application. This breakthrough has sparked a multi-billion-dollar race. Biotechnology companies are now competing to develop safe, effective ways to deliver these reprogramming instructions into the human body without triggering unwanted side effects. Different approaches are currently in development. Some use viral gene therapy to target specific tissues, while others utilize transient mRNA, the same technology used in recent vaccines, to deliver a temporary burst of rejuvenating proteins that naturally dissolve after they've done their work. Startups are even using machine learning to bypass the original Yamanaka factors entirely. By sorting through massive genetic datasets, AI platforms are identifying single gene targets that can reset cellular age without any risk of turning a cell back into a stem cell. At Harvard, researchers led by Dr. David Sinclair are pursuing a different route: chemical cocktails. They have identified combinations of small molecules that can be administered systemically. These chemicals work by opening the cell's internal structures to reset the biological clock. In lab tests, these specific cocktails successfully reduced the biological age of human cells by more than 3 years in just 96 hours. This shift towards scalable mRNA and affordable chemical pills marks the point where age reversal transitioned from a genetic experiment into a versatile medical toolkit. This technology is no longer limited to petri dishes. In early 2026, the FDA cleared the first human clinical trial for a partial epigenetic reprogramming therapy, developed by a company called Life Biosciences. The study targets severe age-related vision loss. The eye is an ideal testing ground because it allows for localized delivery and extremely precise measurements of how well the therapy is restoring visual function. The trial also includes a sophisticated safety mechanism. The rejuvenation genes remain inactive until the patient takes an oral antibiotic pill, which acts as a switch. If side effects are detected, the patient stops taking the pill, immediately halting the reprogramming process inside the retinal cells. With this study, the attempt to restore youthful cell function in patients has officially moved from the lab into the clinic. Global regulators are already preparing for this shift. The FDA has introduced new, flexible frameworks designed to accelerate these mechanism-based therapies, recognizing that aging is the primary driver of most chronic diseases. If successful in the eye, the same logic will be applied elsewhere. Ongoing research suggests we may soon be able to use partial reprogramming to reverse neurodegeneration in the brain, regenerate heart tissue after an injury, and even restore immune system function. While early treatments may be expensive, the move towards scalable mRNA and chemical pills suggests these therapies will eventually be democratized. That have every reason to lower the trillion-dollar economic burden caused by age-related decline. Instead of managing the downstream symptoms of old age, medicine is moving towards correcting its underlying biological cause. If these trials succeed, extending the human health span will move from a lab curiosity to a standard of medical care.